Yes, sir. Thank you, sir.
Okay. Okay, now we're going to look at how the shift happens. I told you the shift happens because of shuffling of different segments or fragments of the influenza virus genome, which has 8 fragments. And when the same cell gets infected with 2 different viruses, the shuffling can happen. I've already told you that, and we discussed that. Now we want to look at it a little bit more carefully as to where the shuffling really happens, and we'll go to it in a bit. Okay. So that is the same thing I have here. Where do the new H and N configurations, where do they come from? So like I've told you before, there are 18 types of hemagglutinin. Many of them circulate, all of them circulate in birds, like about half of them circulate in humans. I have some in bold. You don't have to memorize any of this again, not these numbers. So I have some numbers in bold which I included maybe this year or last year. Those configurations have affected humans only in the last couple of years. So those are in bold. So the reason I have that in bold is that you understand that the H and the N configurations that affect humans are constantly expanding. What about pigs? So what about pigs? We say bird flu, we say swine flu. What is the role of pigs? So pigs can be infected by avian strains as well as human strains. That makes them the ideal hosts for or ideal mixing vessels for strains that come from birds or strains that come from human beings. Both of them can infect pigs, and you remember the reassortment cartoon, like shuffling of cards. This genome that comes from humans and the the virus genome that comes from humans and the virus genome that comes from birds, they can get reassorted in pigs, and pigs are called mixing vessels. And in pigs, it happens more in the digestive tract. Pigs are very susceptible to influenza virus infection. However, the mortality rate in pigs is very low. They don't usually die because of influenza virus infection. They survive. I mean, in general, mortality is very low in influenza viruses, whereas in birds and humans, it's fairly high. Okay, so in the pig's digestive tract, these viruses from humans and birds can get shuffled, and they could be shed in the feces. Now, how is a new configuration of H and N generated for a new pandemic? Here's a quick look. You don't, again, it's just like a conceptual note. What does this say here? Maybe we'll follow this track right here first. So avian influenza virus was replicating in birds, as the name suggests, and this was happening for several centuries. And the antigenic drift or reassortment among birds, or were pigs involved, we don't know. But the reassortment within birds allowed the generation of a H1N1 strain. It's either mutations or reassortment. And a H1N1 strain was generated that in 1917, 1918 that could infect humans. And that was the biggest pandemic that we know of in terms of influenza virus. And that happened in 1918. Now, so after that, what could happen? How did the other strains come up? Possibly like this: human strains and bird strains infect pig. You can see the blue and the yellow virus, and it gives a mixture of the blue and the yellow virus. This could affect both birds and humans. So pigs serve as the mixing vessel, or the reassortment primarily happens in pigs because they're susceptible to both the bird viruses as well as human viruses. Okay. Then I told you that the reassortment is primarily in influenza virus A and not in B and C. We also don't see pandemics in influenza virus B and C. Anybody wants to take a guess as to why this could be?
So again, possibly because reassortment is not taking place.
That's
So
right. The reassortment is not taking place. You're right. So it's primarily because there are no animal reservoirs. You get that? There are no animal reservoirs. At least there is one exception to this rule. I'll tell you by the end of today's class, hopefully. But in general, you can take it as no animal reservoirs. So if you don't have the animal reservoirs, everything comes from humans, and you don't have the new H and N configurations for these. for the shuffling, everything is one host, then the shuffling is less because it will be only drift and there's very little chance of shift or very little chance of reassortment. So that's why it's primarily only in humans. So that's why there are no pandemics with influenza B and C. A little bit about what's going on right now. So this, I think I googled and put this here yesterday or today. I just put this here. So there is an outbreak of bird flu going on in the UK right now. This is only about 5-6 days old. It's happening right now in UK, and many farms were closed. And this is the H5N1 strain that is happening in the birds. Birds are dying. So what do we do if this happens? I'm going to tell you that also in a bit. So this is a current outbreak in UK. What about India and Asia? So in India, Kerala has an outbreak of bird flu, and South Korea has confirmed a H5N1 N8 strain just 2 days ago, Jan 27th this year, 2 days ago. So whenever I do the class, I just Google it and there's always an outbreak. So you understand that some place or the other in the world pretty much has a flu outbreak. Most of the times it has an outbreak. Now, I told you an outbreak is going on in the UK right now, H5N1. What is the problem with H5N1 is that about half of the people who get it, if humans are infected, almost half of the humans, 50% almost succumb to the virus. That's very high mortality rate for this. Whereas H1N1 and many other strains, the mortality rate is less than 1%. This one is 50%. It's very pathogenic. We don't understand why. But almost every other human infected succumbs to it. Now, because this is a dangerous strain, what's happening in UK where they had the outbreak, they are just culling the birds. Okay, so that means culling is just killing them and dumping them with biosafety, all the necessary biosafety precautions. Now, apart from birds and pigs, can other animals be infected? The answer is yes, they can be infected. So, which are the animals? So the animals include cats. There was an outbreak in New York City among cats, a small one, and it can also infect horses. So sorry, not horses, I'm so sorry, dogs. So this is called canine influenza. So these two are the other animals apart from humans, birds, and pigs that have been infected with influenza viruses. Dogs and cats are included in the list. Now, if you eat infected pork, because pigs can be infected, can you get influenza virus if you eat properly cooked pork? It's safe. It doesn't spread by properly cooked pork. But however, the person who's serving, if he has it and if he sneezes on it, and if it's not hot, you may still get it. So that is a different story, but it's not transmitted by food that is fully cooked. And okay, that's all is the idea of this slide. Okay, so everybody with me until this point? Any questions? Anybody? No questions? Are you guys hearing me?
Yes, it's pretty much clear.
Yes, sir.
Okay, let's go on. So we'll move on to influenza virus vaccines. What about influenza virus vaccines? Before we go on to understand what kind of vaccines are used currently, we need to understand a little bit about what are the pandemics we had, what is the configuration of H and N in those pandemics. So like I told you, the 1918 pandemic, which lasted about 2 to 3 years, is the biggest pandemic with influenza virus, is among one of the biggest pandemics that mankind has seen. So this is called Spanish influenza, and there were more than 100 million deaths in the world that time. So we had another one about 60, 70 years ago called the Asian flu, primarily occurred in Asia, much fewer deaths. And there was another one in the 1960s that was even more fewer. But these two occurred primarily in Asia, but this was a real pandemic that hit the worldwide. But what I want you to focus on is that the configuration of the H and the N, at least there is a subtle difference every time. H1, H2, H3. You can see that there is a difference there at least. So, so usually these pandemics or large epidemics are associated with different H and N configurations. So here's a slide that gives you a slightly better understanding of the pandemic strain. So first time it happened in the 1910s or around 1917 this started. So that time actually it happened from a bird to human being, and then it spread across the world and took 100 million lives. Okay, so this one, we don't understand this that much, but we think actually we just use it as a reference point. It is most likely people think it's antigenic drift among birds that caused this. We don't know what is the actual reason of this. This virus is a bird virus that acquired the ability to infect humans in around 1917, and then this swept across the world. Now, that is this green virus is used as the starting point, and that's the H1N1 that caused the biggest outbreak. Now, the second outbreak. So if you look at this, this is same as this virus that was circulating after the outbreak in small numbers across the human population. This is the H1N1 from 1918. Some minor mutations would have occurred in humans, but it's the same H and N configuration. Now, this is the H2N2, which is an avian virus, and reassortment happened where, and most likely, where did the reassortment happen?
In pigs.
In pigs. And what you have here is you have primarily a green virus, but you have 3 fragments of red, or 3 fragments from the bird virus. Now, what are these 3 fragments? These 3 fragments include hemagglutinin, neuraminidase, and the polymerase. So these are the 3 fragments that were shuffled, and this is the virus that was able to infect and cause a large number of infections, primarily in Asia, and caused about 2 million deaths. This happened much later. Now look at this virus, which is the green and the red virus. That is the same as this virus. Okay, that's circulating among humans for the next 10 years. Before there was a reassortment with the avian virus, which is the yellow virus here. Now, this virus is the base for this new virus. You have 2 yellow fragments, and what are those fragments? Hemagglutinin and the polymerase. So it has a new H configuration. That's why you have the, instead of H2, it becomes H3 because it has a yellow H fragment which came from the bird virus, which is H3. So this is the new configuration. So we don't know what's going to be the next configuration. That could be anybody's guess. Talking about guesses, what I have here is what kind of vaccine is used. So this is the most widely used vaccine for influenza virus. It's called an inactivated or killed influenza virus vaccine. This is what most of us get. If any of you are vaccinated, most likely you got this one. So what is the configuration of this vaccine? For a long time, until about 2015, we had something called the trivalent vaccine. It's probably still there, but there is a slight improvement to this in the last 5-6 years. I'll tell you what that is also. So there are two types of influenza A, which is H1N1 and H3N2. You can understand where it's coming from. This is the last pandemic, and this is the most widely circulating strain still. So you have H1N1 and H3N2. So those are the constituents of influenza A. We also include influenza B in that vaccine, a killed influenza type B virus, which is not A. It is influenza B, which does not usually have animal reservoirs, but it causes less severe infection, but it's very common among children less than 5 years of age to protect them. This is also included. So it's called a trivalent vaccine because it has 2 configurations of influenza A and 1 configuration of influenza B. Now, it has an efficacy of about 70%. More or less same for Covaxin. If you look at COVID-19, somewhere in this ballpark. The efficacy is slightly lower among older people because they have a slightly compromised immune system. There is also another alternative to the killed vaccine. It's called the live attenuated influenza virus vaccine. Pretty much the same configuration as this. The configuration does not change, What changes is that this one is completely killed or inactivated, while the second option is a live attenuated or a weakened vaccine. And like I've told you before, if you want to give a live attenuated vaccine, just remember polio virus, you understand live attenuated and killed vaccines very well. Now, oral polio drops, they put it in the route that it normally enters if it's live attenuated. If it's a killed poliovirus vaccine, they just inject it. Same principle for anything that you can think of. Most viruses, at least respiratory and diarrheal viruses, this principle will be perfectly working. Any vaccine that's killed vaccine is injected. Any vaccine that's live attenuated is usually you try and give it in the way that the virus natural infection occurs. That is the route of administration of the live attenuated vaccine, because you want it to go and infect some tissue which it normally does without causing disease. So you want to target that route of inoculation because you want the virus to replicate. It's weakened virus. You want it to replicate. So this is a nasal spray. They just cover your nose and then you just spray a mist and you're vaccinated. You inhale the mist, you're good. So you're vaccinated. This is actually usually done in Western countries for doctors and nurses who come in contact with patients, because it's believed that, anybody wants to guess why that may be helpful for them?
Because they're more susceptible to it.
They are more susceptible, but how does this help? That's correct answer. But how does this help? How would this be better than this? Maybe you all know it. What is the one major advantage of live attenuated? Guys?
Stronger
It can
immunity.
replicate.
The local immunity.
That's correct. It induces local immunity or IgA antibodies. So if a doctor or a nurse took this nasal spray, they would have IgA antibodies in their respiratory tract that will neutralize the virus then and there without letting it get into the blood. Whereas the killed vaccine, it will only stop the virus once it enters the blood. So there will be some amount of damage. But of course, there's enhanced immunity locally also, but it won't stop the virus from entering. So this is actually way better in terms of, if you look at it like that, there is a better chance of stopping infection if you use this vaccine. So that's why healthcare workers are administered the flu mist or the live attenuated vaccine. The reason it's better is because it boosts local immunity. The disadvantage with this class of vaccine, as it is with live oral polio vaccine also, is that it cannot be given to certain groups, including immunocompromised groups. This is not true for oral polio vaccine, but here it's true because some children, because respiratory tract for some children who have pre-existing respiratory conditions, it may be dangerous to give a live attenuated vaccine, so they are not given, they are given a killed vaccine. Now, how are these flu shots made? Okay, so I told you in the diagnostics part that influenza virus can grow very well in eggs. So most of these vaccines are egg-based vaccine. So they use millions of eggs to make the influenza virus vaccine year after year because it grows well in the eggs. So some people have egg allergies. So for them, actually, there are alternatives where this virus can be grown in a cell culture and then killed and used. So that is both of these are for the killed vaccines, growing a lot of it in eggs or in cell culture, killing it, and then injecting it. Now, what about the live attenuated vaccine, which is the flu mist? You just inhale and you get vaccinated. How is the attenuation done? Remember, the attenuation for the polio virus is done in kidney cells, forcing the virus to grow in kidney cells. That's the way it's done. But here, how is it done? They have a strain which is cold adapted strain, or this strain of influenza virus can grow only at 33 degrees centigrade. It cannot grow at 37 degrees centigrade. Now, this strain is infected with H1N1 or the H3N2 strain, and then when they replicate in the same cell line or within eggs, some viruses will have this configuration. What is this configuration? Everything comes from the cold adapted strain except the H and the N that comes from the strain of your choice. It could be H1N1, it could be H3N2, it could be another one next year. WHO will recommend another one. It could be that. So when these two are put into the same cells, there would be at least a few viruses of this configuration which you can screen for, and then you can purify this virus, and then you can start growing this, because all these light gray fragments do make sure that this virus cannot replicate at 37 degrees. This becomes attenuated. Why is that attenuation? Because this virus can, it's a live virus, it's just weakened. It's just weakened because it can grow only in the upper respiratory tract, which is the nose and the sinuses. It won't be able to infect the lower respiratory tract because the temperature here is slightly lower. It grows only in the upper respiratory tract, or is able to infect the cells there and replicate there, but not in the lower respiratory tract where it can actually cause disease. So that is the attenuation of this virus. So what about some new developments? I told you that the trivalent vaccine with 2 influenza A configurations plus 1 influenza B was the tradition until about 2015. But in the last 5-6 years, we have something called a quadrivalent vaccine with 4 strains. So 2 strains of influenza virus A, and that will be 2 H and N configurations, H1N1 right now and H3N2. So these are the ones from influenza A. There are 2 variants. So scientists later in 2012, there was a paper where they emphasized that there are 2 different lineages, not H and N configuration, but very different strains of influenza virus B, one from Asia, one from America, and there is less cross protection between them. So both of them are included in this vaccine. So 2 influenza virus A plus 2 influenza virus B makes the quadrivalent vaccines, and this is what we get today. Now, how do you treat anybody with an influenza virus vaccine? If you're a healthy person, you have mild symptoms, it's just supportive treatment. You take paracetamol or some fever medication that'll reduce your fever. That's about it. Nothing much else. You'll recover. But if you are a high risk, or if you're developing more lower respiratory tract symptoms, or if you're a child under 5 years of age or somebody above 65 years of age, they may be given antiviral treatments. So antivirals are not used for most viruses, like for example, polio or enteroviruses don't have any antivirals ever available. Nothing works against them that we have right now. But there are only a handful of viruses where antiviral therapy is used. So among them, the ones that cause acute infection, very few of them are treated with antivirals among acute infecting viruses, which don't cause which the body clears in a week or 2 weeks. They're usually not treated with antivirals. Influenza virus is an exception because it can be life-threatening. There are 2 or 3 antivirals. 2 of them, the first 2 here, amantadine and remantadine, target the M2. How M2 protein? What is the function of the M2 protein? Anybody recalls? Guys?
Sir, uncoating of virus in the endosome.
Uncoating of the virus. Okay. So it's okay, that is the M2 protein. How do these M2 inhibitors work? These 2, amantadine and remantadine, how do they work? They go into this channel and they bind to the spore. So this is ion channel which selectively allows H plus ions, right? I told you that proton ion channel. So proton channel. So this amantadine and remantadine go into this channel, and they just bind here and block the entry of H plus ions because they already inside the pore and they block it. So that is the way they work. So they prevent uncoating, and that's the way M2 inhibitors work. There's a flip side to it. These 2 M2 inhibitors, amantadine and remantadine, are associated with high levels of resistance, which means that, like, if you start giving it within a week, the patient may develop resistant strains, or resistant strains may already be circulating in the world. So these 2 are now not so widely used because there is a high level of resistance. They use something called a neuraminidase inhibitor. You might have heard of this word, Tamiflu or oseltamivir. It is a neuraminidase inhibitor. I have a cartoon as to how it will work. Let's go to this part. Everybody clear until this point?
Yes, sir.
Yes, sir.
Good, okay. This is a cartoon about the neuraminidase. This not only tells you about how the inhibitors work, but it also tells you an important function of neuraminidase that I told you I'll tell you later. Remember when I introduced hemagglutinin and neuraminidase, I told you one of the important functions of neuraminidase is that it helps negotiate the mucin layer by reducing the viscosity. Otherwise, the virus will get trapped and expelled. But it makes the mucus less viscous so that there will be less trapping. So that's one of the functions of neuraminidase, right? The N of the H and the N, this is the function of the N that we already spoke about. There's one more function of the N that you should understand, and that is in the top part of the cartoon. What is this function related to? So this is a host cell, infected respiratory epithelial cell. Influenza virus is replicating inside this host cell. And there are these 8 segments of influenza virus. This is the matrix protein in blue that's helping assembly. And all the 8 proteins gets packaged. So this matrix protein on the outside, remember, it has high affinity for viral glycoproteins, which are the H and the N, or the hemagglutinin and the neuraminidase. So when the budding happens, when the virus is trying to come out of an infected host cell, it pinches off a part of the membrane, and that membrane already has hemagglutinin and neuraminidase sitting there and waiting there. So when this budding happens and it's just about to leave the host cell and to try and go and infect a new cell, there is an important function of the neuraminidase. So neuraminidase, what is the chemical function of neuraminidase? Anybody recalls from the first lecture of influenza viruses? Guys,
Sialic acid.
it cleaves sialic acid. Very good. It's a sialidase which can cleave sialic acid. So here is a receptor for here's the sialic acid on the cell, and the hemagglutinin can go and get bound to it and get stuck. So this neuraminidase is the one that can cleave it and release this virus. You understand what it does? It can actually cleave the virus from its receptor. That's what neuraminidase can do. So this also has other implications. So what is another implication of this? If you think about it more carefully, what happens when the virus gets in? It uses the same receptor. Here is hemagglutinin, and here is the sialic acid. They bind, and neuraminidase may cleave it, so virus may not infect us. So this actually happens during the virus entry. Neuraminidase may affect virus entry. So studies have shown that the entry happens so quickly that, like, if there are, it may be inefficient. Maybe if there are like 100 viruses, one of the viruses may manage to get in before the neuraminidase can cleave off the sialic acid and release the virus. So it has to be really quick. So it has to happen in a few seconds or a few minutes before this cleavage can actually just cleave off the hemagglutinin from its receptor. So this happens even during entry if you think about it more carefully. But but all it takes is just one virus to infect a cell, then it can just replicate and cause the infection and the disease. So even if it's inefficient, if 100 viruses enter our body or 1,000 viruses enter our respiratory tract, if 1 or 2 of them manage to do this, that's enough. Even if it's inefficient, that's good enough. Now, if this cleavage doesn't happen, what's going to happen is these new influenza viruses that are made from an infected respiratory epithelial cell, they tend to get stuck on the surface. And when they get stuck, there is less virus to go and infect the new cell, and the burden on the host goes down. So that is why the function of the neuraminidase is very important, that it has to cleave it so that it allows the virus to be released and go and infect the next host cell. Okay, now what will happen when you use neuraminidase inhibitors? So here, the orange colored spikes are the neuraminidase. Same here. So these are the neuraminidase inhibitors, Tamiflu, oseltamivir. There are other names for it, just one compound. So what they do is they are able to go and bind to the neuraminidase. They are actually sialic acid mimics. If that will make it easier, they homologs or mimics of sialic acid. They look like that, and they go and block the neuraminidase, and they prevent it from cleaving the hemagglutinin from the sialic acid. So when you use this neuraminidase inhibitors, a lot of virions are unable to be released from infected cell, thereby allowing the immune system to easily handle it. Because one infected cell, remember, can release like 10,000 to 100,000 viruses it can release. If that is contained big time, the burden in your body or the burden on your immune system goes down significantly, and that reduces the severity of disease, and that's how these neuraminidase inhibitors work. Is that clear to everybody? Do you have any questions?
That's clear.
Okay. Okay, after this, what do I have? Okay, so this does not prevent infection. Neuraminidase inhibitors will not really kill the virus, but they prevent the number of free virions available to infect new host cells. That is what they reduce. So they eventually reduce the severity of disease. That's what I have here at the bottom of the slide. Now, in terms of precautions and preventive measures for influenza viruses, what do you think should one do? All of you are adept at it, because everybody now, even a layman, knows everything to do about respiratory viruses. What does one have to do? Guys?
Cover your mouth and nose.
Cover your
Don't
mouth and
sneeze on
nose.
people or
That's
cough.
perfect. Okay, that's good. What else?
Constant sanitizing.
Very good. So that's all. Pretty much that's what is there. I mean, all of these things you all know because of COVID. So this is what one of you said: cover your cough or sneeze, wash your hands or sanitize your hands. And these are some things we already know. This vaccination is important. That's important for COVID. That's important for any disease that we can talk about. That's important for influenza viruses. So this is also very important, that you should stay away from work or school if you actually have symptoms that are related to respiratory tract infections. If you don't have a diagnosis also, it's better to have, because influenza viruses, they are more seasonal in nature. So it's a good idea if an outbreak is happening in the city, even in small numbers, if you have symptoms, it's better to isolate yourself and stay away for 5-6 days. This is actually it has a shorter period of infection in the body than does SARS-CoV-2. It's all over in 5-6 days. Everything is controlled very well for influenza viruses in general. So if you don't go to school for a couple of or to work for 3 or 4 days, that takes care of not infecting new people. And surveillance becomes an essential part of the whole thing, that we are doing very well. We are sequencing a variety of strains from all over the world. We are understanding the emergence of variants for SARS-CoV-2. Just like that, influenza virus, actually all our understanding about why we need surveillance. I mean, a lot of things about respiratory virus vaccination and surveillance, the entire understanding that scientists have in the world today comes from influenza virus, because influenza virus has a very good surveillance program. Like the WHO, the World Health Organization, has several laboratories across the world, but there are 5 key laboratories which are referred to as H5 laboratories, which are the key points from where WHO collects data. What kind of data do they collect? They collect genomic sequencing data. Wherever somebody is reporting an influenza virus, or it's reported in animals or humans, it's sequenced. So we know the H and the N configuration. We know if there are unusual mutations. They also test for antiviral sensitivity to see if there is drug resistance. They do actually periodically, they monitor humans to see if, look at antibody levels to see if they are exposed to any new viruses. And these centers are important to prepare diagnostic kits. They isolate the strains that are going to be used for vaccine production. So this is what these 5 laboratories do. And one of them is actually based out of Pune. One of the top 5 laboratories for influenza virus is National Institute of Virology, Pune in India. So that does this work for WHO. They have, as you can see, they have representation all over the world, particularly dense in some parts, but they have representation all over the world. Now, why is this surveillance important for vaccine production? Why is this relevant to influenza virus? because it allows us to make the best possible vaccine. How is this done? So this data is summarized, and a lot of people are behind this huge amount of funding just to keep track of which strains are coming and which is the best configuration of the H and the N to be included in the vaccine. Just for that, that is the primary objective of this. And of course, to monitor emergence of a new pandemic or an epidemic, all these things are covered, but right now it's primarily useful for us as end users in terms of having the best possible vaccine. Now, this vaccine data, which strains to use, which configuration of H and N to use, all this comes from the 5 laboratories across the world, and of course, a lot of smaller laboratories feed into them. So, and then WHO decides which are the 2 configurations that are going to be the best ones to be used in one particular year, and they decide those 2 configurations. Right now, it's H1N1, H3N2, and they start making the vaccine. It takes almost 5 months or 6 months to get this vaccine. Let's say they want to include a new strain, H5N1. What do? So these centers, the 5 reference centers, they isolate these strains, right? Wherever they are, they isolate H5N1, they isolate the strain. Now, if you want to include that in the vaccine, what happens is, this is what happens. They identify some strains, they create something called a seed bank, they inoculate eggs. I told you eggs is where they're produced. They harvest or purify the virus. They optimize the concentration that has to be used. They do some safety testing and sterility testing, and they do some clinical trials in small scale and then larger scale, and then the regulatory agencies review it and they approve it for human use. So vaccines have something called an adjuvant. We won't talk about it now. We'll look at it when we look at vaccines. So this whole process of changing a H and the N configuration in the current vaccine will take almost 5 months. So this is the roadmap for that. And if so, there is a flip side to this. What is the flip side? Let's say I just told you and I showed you some cartoons about an ongoing outbreak in UK with H5N1. So if you want to include it in the vaccine, if WHO decides today, the earliest time we can have a vaccine is sometimes perhaps in June, right? In June 2022. Why is this a problem? This is a problem because by that time it could have swept across the world and it has a high mortality rate. So that is why this could be a problem. WHO will have some other plans, and now there are other types of vaccines that are possible, including mRNA vaccines that may make it faster. But this is the flip side of actually having an outbreak with a high mortality strain. For example, we're all still surviving, and it's possible that even though almost by now I think more than half of the world would have been actually exposed to SARS-CoV-2, there's been only maybe, I mean, a large number of deaths, but nothing compared to the influenza virus H1N1. It's still in a single digit in millions. So that is 100 million. So that's all. So that is possible only because the mortality rate of the current pandemic is much lower. But if you have a strain that is causing 1 in 2 or 1 in 3 people to die or 1 in 5 people to die, that becomes dangerous because by the time you have the vaccine, there would be a lot of damage that's already done. So that is what you have to understand. The time to get to the vaccine is very important in the case of these pandemics, and that has been a bottleneck. We are resolving it very well. I think there have been a lot of new techniques with SARS-CoV-2, at least 2 or 3 new technologies. Entirely first time in humans, 2 or 3 new technologies have been used with SARS-CoV-2 to expedite the vaccine. Now, what about, I have only 1 or 2 more slides, I think, for today's class. What about influenza virus B? I told you it causes milder infection than A. They are in that order. A is the most severe, followed by B, and C is the least. They are in that order. But anybody wants to guess why that could be? Guys, does it mean B came in later or C came in the last? Nobody wants to think and answer that.
Naming is based upon the discoveries.
That is correct. And almost there. What could be the reason? We probably discover what is the most pathogenic to us first. That's why A came first, and then B, and then C. Actually, B is older than A, and there is literature to state that. But we named it in the order of discoveries, like you rightly said, and that is because we only focus on what causes disease. We focus on that. We don't focus on a lot of bacteria in the stomach. There are millions of different types of bacteria that don't cause stomach. We don't even know their names fully well. So we only focus on the ones that cause disease. So A, most pathogenic, followed by B, followed by C. Okay, so that's the degree of disease that they cause also. And a lot of children under 5 years of age are exposed. Usually they recover well, but there is a small amount of mortality, and that's why this is also included in the vaccine. Okay, so some reports indicate that some seals have it. I read this almost like, I don't know, 7, 8 years ago. But after that, there have been any more reports. So this is the one exception to the rule that there is no animal reservoir for influenza virus B. And this is included. 2 strains are now included in the quadruple vaccine for influenza, 2 strains of influenza B. C is the mildest one. There are no major outbreaks, only small numbers, but still a large number of children under 10 years of age are exposed. More than half of us, before we are 10, we are exposed to this virus. Almost all of us are exposed to this virus already. So they cause mild infections. We don't care. It might pass off as a common cold, and there are no animal reservoirs reported for influenza virus C. And that's why it's not included in vaccine, because it's very mild and very small outbreaks. Okay, I think that's all I have in today's class. Okay, we have a few minutes. Anybody has any questions on influenza virus, we can take it.
So,
Yeah, go ahead.
in the case of antigenic shift, suppose someone were to get infected with H1N1, and then because of an antigenic shift, they get infected with H2N1. Would they have partial immunity since N1 is there?
Absolutely correct. So because if you look at the H and the N, I would look at it like this. One, antibodies against the H, if they are subtype specific or configuration specific, they will help you better, because they will even stop the entry as best as they can. They will stop entry of the virus into the cell. Even if a small number of viruses enter, they can't replicate and re-enter and infect a large number of cells. So that protection is usually blocking entry. So like I told you in one of the slides, that antibodies against the neuraminidase don't stop or block infection. They only reduce the severity of disease, because neuraminidase does these 2 things. One is negotiate the mucin layer. Second is actually preventing aggregation on top of the infected host cell. And that allows the numbers to go up. So they would have the second function intact in the case example that you gave, because the N configuration remains the same. They would still have N antibodies. So they are likely to have less severe disease compared to the ones who did not get exposed to H1N1. So N antibodies or antibodies against N, if it's matching the strain that you're currently exposed to, then it helps reduce severity big time. It is really helpful. They may get infected, and they will get infected in the example that you gave, but they are likely to recover with very mild disease.
Okay, thank you, sir.
Sure. Anybody else has any questions?
Sir, I have a curious question.
Please go ahead.
So, as we were discussing, we have, like you were saying that in 1970 the virus got mutated and the different component of the virus mixed, and in 68 the avian component. So today too we have sequencing people come to know how they were coming to conclusion that there is a mixing of virus from different strains earlier, and the way we have some carbon dating system in monuments where we can find out that this carbon amount is this. so we can presume the timeline that this monument is or something is like that much old. And how do we come to know about this timeline for the shift and drift? What made us conclude that the drift happens in like 10 years and the shift happens in 100 years?
Okay, so okay, I'll try and explain that. A lot of questions, but I don't know where exactly to start. But in terms of how do we know, there is nothing like carbon dating. But if you were to, if you ask me how they do it, these are done from samples, archived samples from that era. You sequence it, and you have right now you have several tens of thousands of influenza virus genomes available, and you can construct what is referred to as a phylogenetic tree. And there are ways of calculating and or estimating a time of divergence from phylogenetic tree. This is more theoretical, but proved, I mean, or widely accepted. It will be off, it won't be accurate, but it will give you a good ballpark of when the strain emerged from other influenza viruses or which is the closest relative. All these things can be tracked by using full-length genome sequences and good phylogenetic tree and analysis of branches and distances for divergence. This can be analyzed. So in terms of understanding how do we know it's drift or shift? Maybe I wish, maybe can I, how many minutes do we have? We have some time. Let's see if I can quickly share my slides again and go back to one slide where I can try and explain this to you better. Give me just 1 second. Okay, here we go. This is the best slide. Okay. Now, if you had samples from 1918 pandemic, which we do, and we've sequenced them, they look like this. Whatever they look like this. Okay, let's just put it as baseline. Then they have samples from this one. And what they find is this one does not have mutations, but the entire thing is like very different. Like it's like 50% or 60% different from this fragment. This green and this red are so different, it is not possible to happen by mutations. We know that. And before that itself, we knew that the shuffling can happen and all that. So when this is so different, we know this is a new fragment. Now, when you sequence this virus, this green and this green will match. Even if they are 1% different or 2% different, they're going to be a close match. Same for all the greens in these two viruses here. Okay, I think I don't have the pointer. Give me a second. Okay, in this green here and this, all the greens here are going to be very close matches with 1 or 2% different at best. But these three reds are going to be like 30, 40, 50% different from these greens. So we know that that probably came from reassortment. Does that make sense?
Yes, sir. Yes, sir.
Okay, and if you look at this, the same thing. This red and this red will be more or less identical with subtle differences. The rest of the greens are going to be a match to this greens with subtle differences. But these two yellows are going to stand out. They're going to be like that, like let's say approximately 40-50% difference. That may be actually close to a real number of in terms of sequence identity or sequence similarity between this red and this yellow. This red and this yellow may be very different in terms of sequence, so we know that that is different. So we also have avian samples from that time, because by that time we became good in archiving. There are very few samples from the 1918 pandemic, but there are samples. But by the 1950s, scientists became adept and they understood the value. So there are avian samples available from this era, and now we have a lot more avian samples. Like every bird that dies in a poultry farm, sampled, fully sequenced by the respective WHO reference laboratory, archived. So we have a lot of information that will allow us to know that if a fully red virus from birds existed at that time, we have samples for that. This also we have samples for this. And now today's world, we have all the possible samples. Every bird that dies, sequenced. The sample is amplified and it's fully sequenced. Does that answer your question?
Yes, sir. Thank you so much.
Yeah. Okay. Anybody else? Any other questions? No other questions?
Sir, for sequencing of birds, which kinds of sequencing are being used nowadays?
Okay.
Like, so
Which
if you are
kind of
telling every bird dies and we are going to sequence, so for that we need lots of money also, right?
yeah. So this is not a bird in the wild, it's a bird in a poultry farm. You get the difference. Some, like in a forest, if a bird dies, we don't do anything, right? But if in a poultry farm where we have like tens of thousands of birds or millions of birds, like the one that's happened in UK right now, they had about 1 million birds and one bird died, and they took a sample and quickly they found out H5N1. All the birds are killed. That sample is archived. So this is done in India also. There was one outbreak in Kerala. This is only 3 weeks ago. I'm just talking about recent ones. They keep happening year after year in all the places. So this Kerala, when the bird died, they took a sample and sequenced. I can't remember the subtype, but they sequenced it. They culled the birds. So this is what happens when a bird in a poultry farm dies prematurely, but not for anything in the forest or anything on the road and all that. We don't care, but every bird in a poultry farm. Does that make sense?
Yes, sir. Like in UK, they are sequencing every child, so they may have available that kind of technology and that kind of system. But do we? Is there in India also we have such systems for sequencing?
Yeah. If you give that sample to me, I can sequence it and tell you the result tomorrow in my lab itself. So India is not so
Yes,
bad.
sir. Sir, do you have sequencer?
We have actually two. We have a second generation sequencer. We have a third generation sequencer, wherein you can sequence without knowing what you're sequencing. You can sequence any sample, any DNA. You can get the sequence and assemble it. We do sequencing routinely. Yeah.
Sir, which sequencer have like Illumina or Oxford Nanopore something?
We have both actually. We have a MySeq from Illumina and we have a GridION and a MinION from Oxford Nanopore or ONT. We have actually 3 sequencers.
Okay, sir. Okay.
Sure.
Sir, I want to like see someday I will come to see.
Sure. You're very welcome. Actually, if anybody else wants, actually, or maybe if you want, maybe we can do a demo if things get better in the second half of the course. We can perhaps have a demo on how these sequencers work. I will try to do that.
Okay, sir. Thank you, sir.
Sure. Okay, I think we are overshooting time. I think we have 1 minute left. Okay, guys. Thank you very much. I'll send you the slides. Bye-bye.
Thank you, sir.
Thank you, sir.
Bye.
Thank you, sir.